Hello, nice to meet you. A pleasure to talk a little bit about the biodiesel, green diesel, and the sustainable aviation fuel at the Biofuture Summit. Thank you very much for the invitation of BBEST. That originally was scheduled to the last year, 2020. And a pleasure to be with you now, talking about these issues. I'm professor at Federal University of Rio de Janeiro in Brazil. And also, I'm very close to the biofuel industry, mainly the biodiesel and the biojet fuel industry in Brazil, as a technical consultant of UBRABio. UBRABio is the largest association involved with biodiesel and biojet down here in Brazil. So I'm in close contact with the industries, and let's talk not just from the perspective of academic level, but also industrial level, because I'm very involved with those people, right? A little bit about the current Brazilian biodiesel scenario. You see now a map with the Brazilian map, and you see the green points are the plants, the biodiesel plants that you have. The first plant in Brazil, we started in 2005, so very recently. And now we have about almost 50 plants, 49 plants in 14 different states. We have biodiesel plants in all the regions in Brazil, in the south, and the north, and the west central, and the southeast and northeast. We have a very spread amount of biodiesel plants, which is different compared to the petroleum refinery, which is very close to the coast, to the ocean, right? That's not the case of the biodiesel. Well, so far, the total amount of produced and consumed biodiesel has been about 50 billion liters, 50 billion liters since 2005, when we started the first plant. And with the 2021 March, approximately, roughly 50 billion liters so far. This means approximately about 95 millions of avoided CO2 emission, which is a lot, almost 100 millions of metric tons of avoided CO2. This is ascribed just for biodiesel. As you know, we have also an ethanol program, a huge program in Brazil, but just dedicated from the biodiesel, almost 100 million metric tons of avoided CO2. It's a lot. It's a so huge amount of CO2, which is even difficult to imagine what does it mean. One of the ways that we're using for to have an idea about what does it mean is 95 million of avoided CO2. We can talk about the equivalent reforestation. I can explain better. If you don't have the biodiesel program in Brazil, zero biodiesel production, and instead, if you have a reforestation program, we would need about 693 million trees, which would be the equivalent of CO2, avoided CO2. So now it's much more clear. In terms of area, we would need to make this huge reforestation program instead of the biodiesel program, almost 48,000 square kilometers. Just to have an idea, the Rio de Janeiro state, it has 43,000 square kilometers. So we would need more than the Rio de Janeiro state area to do this reforestation. So it's a huge program with a huge impact in the CO2 emission. And look, that we're talking about a young, relatively young program compared to the ethanol that we have started in the 70s. In biodiesel program, we have approximately 15 years. So it's a big impact, absolutely. We have started this program with mandatory blending of B2 in 2008. And later on, we have moved to B3, B4, and you can see the current law that you have is guaranteed to achieve B15. In 2023, we will have this mandatory blending of B15 in all the diesel in the country. Currently, in 2021, we have the mandatory use of B13, 13% of biodiesel in all diesel, fossil diesel used in the country. This is a law. This is guaranteed, right? And we are working to improve this law to achieve B20 by 2028. And this is one of the goals that you have in our biodiesel policy in Brazil. So we look to have this B20 very soon in the country. In terms of capacity, despite to have a mandatory program with B13, actually, currently, the capacity that you have is close to B17, so have an excess of capacity. And with the new revamps that you have in industrial plants, we're very close in few months to achieve B22 in capacity. So we don't have any problem with the hardware, with the plants, biodiesel plants, the transterification plants that you have. And it is scheduled for this year, 2021, to achieve the prediction of 7 billion liters. It's almost 2 billion gallons a year. And actually would have in capacity more than 10 billion liters capacity to be produced. And also we don't have any problem with raw material, with feedstock. So we're very good in these numbers. It's very interesting when I talk about biodiesel. We have a type of a paradox of cost and value, pricing and value, right? And recently, an official enterprise of research in energy, EPE, is official government company of energy research, and they published a very interesting paper describing the biofuel have impacts in São Paulo metropolitan area, just evaluating the amount of particulate matter, mainly the smaller than 2.5 microns, in the health, because as you know, it is very ascribed to cancer in the lungs, and also a lot of problems with the heart. And due to the biodiesel and ethanol used in Brazil, we have approximately per year, just in São Paulo metropolitan area, avoided 615 deaths a year avoided. It's a lot. And if you move from B10 to B15, as you have in 2 more years, we'll be avoiding 104 more deaths annually. So this is a big impact in the health. That is a very important issue, mainly during this pandemic situation that we're experiencing now. So this is very important to emphasize, because biodiesel is not just an energy fuel, but actually are saving lives using this, because we're not just talking about greenhouse gases, but also about particulate matter, and it means health. So this is very important. This is one of the benefits. There are a lot of other ones. We have zero sulfur, so zero SOx emissions, and we decrease a lot the amount of carbon monoxide, and a lot of other benefits. So we have to discuss very well this paradox of value and pricing. we are convinced that the next step of the biofuel scenario in Brazil will be including green diesel and the called sustainable aviation fuel, SAF. We have visited different plants in the US, in Europe, who have participated in the first commercial flight using biofuels, using SAF recently. And you know very well the situation. We are in favor of this new type of renewable fuel, but it's very clear to us that this has to be used instead of fossil diesel, not in competition against the biodiesel. So this is very clear to us. We're in favor of those new biofuels, and this will be used instead of the fossil ones. Can we produce green diesel and SAF from ethanol? Yes, we can. We can use this dehydration process, the so-called alcohol to jet and alcohol to hydrocarbons to produce that. So this is interesting. You can make some type of integration with the ethanol people in Brazil and in the US, of course. Can we produce green diesel and SAF from biomass? Yes, we can. We can use a huge amount of sugarcane bagasse and other feedstocks, residual ones from agriculture, to do some type of a gasification and Fischer-Tropsch synthesis. It's very interesting. Again, integration issue. And of course, you can use the more novel process to produce green diesel, HVO and SAF, using lipids from vegetable oil and from animal fats. So this will happen here in Brazil, and this will be very, very interesting. However, we have to take into account the following. When we have any type of combustion, we need oxygen, not just the fuel, but also we need some type of carburant, which is the oxygen. And we need oxygen to have a complete combustion. Otherwise, we can have particulate matter that we don't want, as I have explained earlier, right? So this is very clear. In everywhere, for oxygen, for gasoline, for instance, in Brazil are using for decades ethanol. USA are also using ethanol. Europe is using other oxygenated fuel like MTBE, ethyl tetrabutylether, and China are using methanol. This is very important. We need oxygen for a complete combustion, and oxygenated fuel can do that, can promote that, and that's been used for years, for decades, okay? In the case of the diesel engines, the only alternative that you have, it is called biodiesel because of the ester. In the esters, you have the oxygen, like you see in this picture. We don't have this in green diesel or even in the fossil diesel, of course. So even with the new type of biofuel like green diesel, we still need biodiesel, the fatty acid methyl esters, in order to promote these oxygenated molecules, the complete combustion, and the reduction of particulate matter. So this is very important to emphasize. We still need biodiesel. And the final issue that we'd like to talk this workshop, this seminar, is about the biofuel onboard reforming. We see very clear that in more years, we'll have the hydrogen economy. It's very clear to us and very clean as well. Actually, you have to produce hydrogen to be used in a fuel cell like the SOFC. And we're in close contact with Unicamp people involved in this research, or working together with them. And the idea is to transform the hydrogen using the whole infrastructure that you already have for ethanol or biodiesel, and you have for green diesel, and you have to have in the vehicles the biofuel onboard reforming, or using the infrastructure, the logistics of ethanol and other biofuels, and on board you have reforming, producing a green hydrogen and a clean system. So this is a type of a microchannel reactors that we are working in research in the Federal University of Rio. This is the center that I'm coordinator, And we have a lot of students doing that, and very exciting because it's very important to use this type of systems in the ethanol and biofuel reformer, because we need an integration of the thermodynamics involved in the steam reforming, the water gas shift reaction, and all the fuel cell as well. So this type of device, the microchannel reactors, is the device that you have to use. And we're producing that in Federal University of Rio in a research level, of course. So basically, this is the idea. Thank you very much for your attention. And you can send me questions later on. Bye-bye. Thank you very much.
Ladies and gentlemen, hello. I welcome you all to this Biofuture Summit 2 and BBEST 2020 conference, which is going to be held virtually from May 24 to 26, 2021. Let me introduce myself. My name is Dr. Khurram Shahzad. I am working as Associate Professor at the Institute of Chemical Engineering and Technology, University of the Punjab, Pakistan. Our institute is almost have 100 years of history and the pioneer in our country. Today I am going to present on catalytic pyrolysis of Saccharum Munja in a fixed bed reactor. This is the work done by one of my students during his study, and I try to keep the presentation very precise and short and more detailed. You can see on the relevant paper. That would be the extended version of this presentation, because we have the short time. Layout of presentation is the introduction, the materials and methods, results and discussion, summary and conclusions. So the introduction, we all know that the petroleum product deficiencies, and because of some political, social, ecological issues, sustainable biomass turns into a vital source to satisfy our requirements without affecting the interest of future. So biomass is a very vital source to meet our requirements regarding the fuels, and nowadays the biochemicals is very famous and the focused area. So thermochemical, physiochemical, biochemical are the ways to power innovations and the technologies that are converting these innovations into the practical demonstrations, that are the pyrolysis, incineration, gasification, and but of course not limited to these technologies. So pyrolysis is a proven technique for changing over biomass into oils, which can be utilized as the fuel and the chemicals. And numerous investigations have been completed on pyrolysis of biomass, pyrolysis of wheat straw, wheat husk, and with different catalysts. So we selected Saccharum Munja, that is a fine grass mostly found on roadsides, rivers, canal banks, and among a lot of available biomass, Munja is one of the viable option which is available abundantly and cheaply, especially in the Indo-Pak region. And you can see it, you can find it very easily on the places that are not being taken care very well, and there are not much requirements for this biomass to grow. So it is a decent origin of some furniture related items also, and it is also a potential origin of alcoholic drink. It yields a reasonable amount of sugars on dissolution with the acid like sulfuric acid, and fermentable sugars has also been acquired from this. This is the Saccharum Munja you can see here, and you already have seen during your long visits along the roadsides and near the foresty areas, and maybe some of the parks that has not been well taken care of. Materials and methods. Material Saccharum Munja, we got it from the Upper Punjab region, the Kasur region, and we segregated it and washed with the distilled water to remove the dirt, debris, and dried for 2 hours at a temperature of 110 degrees C, so that the excess of the water has been removed from this and it is free from the dirt and debris, and it is then crushed to get the required sizes before using it in our experimentation. And we make different mesh sizes that I am going to present afterward. And these are the operating parameters for our experiments. So we use a batch pyrolysis reactor coupled with the nitrogen, and each batch was consisted of the 7 grams, and the particle size, different mesh size, 40, 50, 60, and 70 mesh size. And the mesh size, of course, the size that has been passed from the respective mesh. And this is the flow rate of the nitrogen, and the temperature of reaction we chosen 460 degrees C, 485 degrees C, and 510 degrees C. And this is the reaction time for each experiment, and this is the temperature of the water that is being used to condense the vapors. This is the approximate analysis and the ultimate analysis. So the Saccharum contains moisture about 8.38% and ash is 5%. So ash content is also lower in this. And there are the good amounts of the carbon and hydrogen, which is the characteristics of the biomass, and low amount of sulfur as well. So probably there are very much chances of the sulfur related contamination, and this is the oxygen. Here you can see the figure of our the reactor, packed bed reactor. This is the external casing, and these are the connections for the heater. And this is the batch reactor which is being inserted into this heater assembly, and which is surrounded by the proper insulation material. And this is the thermocouple you can see here inserted in at the top of the reactor. This point, this is the thermocouple that has been inserted into this. So we use the biomass with the catalyst, and for the catalyst purpose, the one catalyst we take it from the market, that is ZSM5. Some of the specifications of this catalyst I have put here: cation, crystallinity, surface area, and so. And afterward, we impregnated this catalyst with the copper sulfate solution to mix it with the copper actually. But we impregnated the copper into this catalyst to enhance the bio oil yield and to particularly increase the selectivity towards the aromatics. This is the schematic diagram of our experiment. Sorry. This is the reactor you can see here, and the water tank used to condense the vapors, nitrogen cylinder, and the temperature controller along with the thermocouple and so on. Results and discussion. So effect of particle size on the yield of bio oil. So we use the 5 mesh sizes, 40, 50, 60, 70, and 80 mesh sizes. And at temperature of 510 degrees C, you can see the different values of the oil, gas, and vapors we got during our experiments. And the 40 mesh size, we found the optimum during our experiments. When we check it regarding the size among different mesh sizes, so we get the maximum yield of at 40 mesh size. You can see here, this is almost 25. And let me clear that this oil yield is before equalizing all the parameters. And after equalizing, it will go up to around 35 to 40% when we equalize it to make the taking effect of the other parameters on the Excel sheet when we equalize it. So this is the same experimentation of the different lysases but at different temperature. It is at 485 degrees Celsius and we have somewhat higher yields of the oil, and this one is for the gas about 55%, and the gas yields are found to be maximum for the 60 mesh sizes. So of course the mesh size, the mesh size it matters because it is responsible for the different diffusions, and afterward that diffusions lead to the certain reactions. So if the size of the particle is changing, the diffusion of different gases is changing, of course, to get to the base molecules, and afterward the reactions are changing, different chemistry of the reaction is changing, and that's why we have different yields for them. This is the for 60 degrees C, again the graph of different mesh sizes. So this is the graphs of the XRD which we got after we impregnated the copper sulfate in the form of copper sulfate. We impregnated copper on the ZSM5 catalyst which we got from the market. And if you see, this is the pure catalyst and this is our impregnated version of catalyst. And to better look into it, you can see here, this is the pure catalyst, this is the impregnated catalyst, and we can see here the certain diffractions that ensure us that the impregnation is very well before you can use it. We are also planning to further analysis of these prepared catalysts like BET surface area and other thermal characteristics and so on. So the catalyst to biomass ratio we used from 0, 1, 2, 3, 4, 5, and 5 means 5% catalyst to biomass ratio. So we got the optimum results for 3%. 3% catalyst to biomass ratio that gave us up to 42% oil yield. And the other things you can see for the other parameters, like for the 1%, it was around 36%. And these are the unsaturated. So these are the gas yields, and from here we can see that the lowest gas yield was found at 40 mesh size. So the 40 mesh size, the gas yields are lowest, and when it is lowest, the most probably the oil yields are maximum. And the different mesh sizes, we can see the different values of the gas yields for this particular case. Oil yields we can see here, and again for different mesh sizes, for different temperatures, 510, 485, 460, and maximum oil yield was found at 40 mesh size. These are the charge yields, And this is the graph of GCMS. And here we can see the different compounds we found in our sample, like toluene, furfural, phenol, and that was the objective of our catalytic pyrolysis, that we want to selectively increase the yield of the aromatics. So I am running short of time. So the conclusions. So 0.4% was the ratio, optimum ratio for copper sulfate and HSZN, and 3 for the catalyst to biomass ratio. And bio-oil yield was optimum at 40 mesh size and at 460 degrees C, and most of the chemicals found were the aromatic origin. Thank you.
It's a pleasure to be here in the BBEST 2021. The title of this presentation is On the Way to Obtain Renewable Jet Fuel, Kerosene, Diesel, and Hydrocarbons. I'm Rubens Maciel, professor at State University of Campinas, School of Chemical Engineering. The main motivation of this presentation is the fact that the society requires energy and fuels to live well. The two graphics on the top of this slide shows that there is no country in the world that have both high human development index and low energy yields. At the same time, if you look at the bottom part of this slide, you can see the total energy consumption and the CO2 emissions that comes from the fuel combustion. And then there is an increase, large increase in CO2 emissions. It means that you need to look for solutions in terms of total energy and the energy production and including fuels or biofuels to reduce the CO2 emissions. Then the reduction of the CO2 emission is more than a target, it's an issue, especially in the transport. That is an important fraction of this CO2 emission worldwide speaking. And there is some immediate applications into well-established markets that for biofuels and renewable fuels. That's like mentioned in this case, the bioethanol that from sugarcane or corn, especially for the large amount of it produced in Brazil and US, respectively. The renewable gasoline that can be used also for light cars, biobutanol for light cars, additives for bioethanol use in heavy engines, that means the cycle engines, biodiesel and renewable diesel for diesel cycle engines or heavy engines, and the biokerosene, renewable fuels for jet fuels. Also, we have to take into account the renewable fuels for maritime use and the production of hydrogen, especially for using fuel cells, especially if this comes from renewable sources. And I would like to point out the importance of these biofuels coming together, the biorefinery and biorefinery concept that allows you to produce the commodities as the biofuels are, and the high added chemical values to get energy in the combined cycle that is important for the society anyway. I like to make this comparison between the use of electricity, biofuels, and chemicals, especially nowadays that there is a lot of concern, a lot of interest in the electrification, the transport. That's a good issue, but we have to take into account that in spite of this part of the electricity, for instance, can be produced in photovoltaic panels with a very, very good efficiency, in perhaps 10% take into account of the day or 24 hours. To use it, you have to store this energy, and usually compared to the liquid fuels, that liquid fuels have a high energy content, and they appear to be much more convenient for a vehicle than electricity. Once you have to store in heavy and relatively expensive, even nowadays, batteries, the density of energy of batteries are very low compared to the content of energy in biofuels. Despite of to have to, of course, increase the efficiency of the engines using biofuels. For aviation, nowadays, for practical reasons, only liquid biofuels is possible, at least so far, and that means that we have to focus a lot in the aviation biofuels. Maritime biofuels is also a concern to reduce the CO2 emission, and as I mentioned to you, chemicals, they need to be together, including the possibility of biorefinery that brings more flexibility in the business strategy due to the risk of feedstock price fluctuation due to the intensive use. Regarding batteries, that is quite important point to be analyzed compared to the liquid fuels in the electrification. If you look to this graphically here, you can see the x-axis, the energy density, the y-axis, the output, the density of energy, both are in watts per hour per kilogram. You see that the lithium-ion batteries are around 10 in terms of watt hours per kilogram, compared to the gasoline that's 10 to 6. In red, I put down the E2 oxygen or lithium batteries. They are talking about 10 to 3, so relatively much more larger energy density, but a lot of effort has to be made in the research to become this kind of battery still competitive with or in good shape to be used. So liquid biofuels appears to have a relatively long life in spite of the advances of the batteries. There's several possible routes to produce biomass in products including biofuels. One of them is the first generation that you use straight away the sugar, extract from sugar-rate crops and go to fermentation. And then there's several options. And there are several options to convert biomass in biofuels, including hydrolysis, gasification, and then we'll go to syngas and syngas to biofuels, digestion, to the bacteria go to biogas, pyrolysis, there's a chemical route to bio-oil, the extraction and so on, and the catalytic conversion of ethanol to hydrocarbons, use ethanol then as a feedstock for another materials, or other fuels. Ethanol as hydrogen carrier as a niche to use in fuel cells, and the use of vegetable oils or bio-oil and co-processing or vegetable oil and the oil in transesterification and esterification reaction to produce biodiesel. The thermochemical route is interesting to point out that depending on the operating condition, can go to pyrolysis and get to bio-oil, the left-hand side of this slide. The right-hand side is gasification. You can have the syngas, that's the priority, and then the relation of the hydrogen and CO, this is named syngas. And this you can synthesize then the biochemical route and using the lignin cellulosic biomass have as steps the pre-treatment, the hydrolysis to deliver the sugars that are fermentable, go to fermentation, and then you get to bioproducts, for instance, bioethanol. For the thermochemical platform, you can go to the bio-oil gasification. If you go to gasification, you get to clean up and condition the gas to the catalytic conversion, and again you go to the, for instance, through the Fischer-Tropsch process. You can go from syngas to hydrocarbons. And also there is a hybrid platform in which you can use the syngas to go straight away to fermentation and get ethanol and other products. Bio-oil conversion, that's another possible way to use the thermochemical route. We can split it into two possible options. One of them is using this bio-oil or the vegetable oil or micro oil in the co-processing concept. That means that you can use the existing facilities in the oil refineries to process together oil, and then you can the fractions of renewables, fraction diesel, fraction gasoline, fraction jet fuels. A point of concern is to measure the amount of carbon that these exit streams that are from renewable sources. And otherwise, you can use the biomass to obtain pyrolysis. Through the pyrolysis, you can bio-oil, then you go to conditioning or cleanup steps with a catalytic conversion to obtain green gasoline, green diesel, and jet fuel. And can use also in this thermochemical platform municipal waste as possible feedstock. As I mentioned, we have several options for renewable biofuels. Biodiesel can be blended with regular diesel and be used. Nowadays, we are doing this in several countries around the world. The biodiesel can also be used as a feedstock for jet fuels. They will call the oxygenated biofuels. Also, from vegetable oils and micro oils, you can go to the renewable gasoline, diesel, jet fuel, and hydrocarbons. You may have ethanol for light cars, methanol for light cars or in the fuel cells. The physiotropes synthetic hydrocarbons, gasoline, diesel, and the kerosene, liquefied hydrogen, and the liquefied biomethane, several processes to obtain renewable hydrocarbons, including gasoline, diesel, and the fractions. So, talk about some refining technology pathways to aviation biofuels using sugarcane has been explored so far. You can use the sugarcane and the sugar cane juice and go to ethanol and use catalytic hydrotreatment to obtain hydrocarbons. Or you can use modify this to go to furnace and through catalytic hydrotreatment again go to kerosene, or modify the microbial to the hydrocarbons, catalytic hydrotreatment and go to kerosene. Otherwise, you can use catalytic hydrotreatment to go to hydrocarbon fuels. Another fraction of sugarcane, that means the bagasse or the cellulose or agricultural residues, you can go to the gasification. This means syngas, Fischer-Tropsch, to obtain the hydrocarbons. You can use the fast pyrolysis, get bio-oil or pyrolysis oil, and then this oil go to the refinery for processing, or you do the catalytic conversion, or you go to the biochemical breaking of this sugar in sugars, the minerals, cellulosic material, and go to fermentation again. So, talk about renewable aviation fuels that because it's well established for ethanol and diesel. One in the light engines and not one to have engines. And the renewable aviation will deserve attention once you may have vegetable or bio-oil in Khurram, as I mentioned. The synthetic Fischer-Tropsch kerosene produced from biomass. And they also have the fatty acid methyl ester, FAME, fatty acid methyl ester, PAAA, and the hydrotreatment technology is paraffinic processed ester and fatty acids, the EFA. And of course, the use of hydrogen that is far away to be using nowadays in the aviation market. I'd like to discuss very quickly the one technology that we have developed to the students at Unicamp, that's called Biokerosene, that allows you to produce biojet fuel production in two steps, one of them using vegetable oils or bio-oil. One of them is oxygenated biofuels of high purity with short fatty acid chains. And also allows you to produce the biojet fuel as hydrocarbons. And together with renewable biodiesel, gasoline, and hydrocarbons for different purposes. The advantage that you have the two kinds of fuels that are known as oxygenated hydrocarbons, low cost, high purity, and you can use in airplanes, but not as jet fuel. And you can use the hydrocarbons in integrated holes with very low freezing point. This and less or lower than 47 degrees centigrade. And this product is quite similar to jet fuel. It's a renewable jet fuel. What is interesting to mention is hydrogenation, or a step to withdraw the oxygenation of the molecules as carried out only in a minor fraction of the product, that means low operating costs. And the steps of cyclization and the aromatization is not necessary to be made because usually it occurs during the process that we are using, so no need to have this to promote the generation of compounds of low freezing points. We have had it in our process. And you use intensified process. You can low energy consumption, the fast process. That's a smart size equipment using the concept of intensification process. That means increase too much the heat and mass transfer process. We have, against it, we have to find a way to get the homologation that is required. And the renewable hydrocarbons are not regulated so far. One interesting point about our process, the flexibility. You may use the raw material in all regions of Brazil. So with the same process, it's possible to have the jet fuel or the kerosene or the renewable diesel and gasoline or hydrocarbons using babasu, soybean, castor oil, palm oil, macaúba, and micro oil and bio oil. So depending on the region, you may use the same process to produce what you want. So the process is in a very simple way. You use ethanol and vegetable oil, and then do a reaction step, and then you go to a very sophisticated separation process, but not expensive, and you get the Biokerosene oxygenated oxygenated hydrocarbon, that is right hand side of this figure, and then you have the further reaction to get the Biokerosene and the hydrocarbons, having as a bioproduct biodiesel, green gasoline, green diesel, and green ethylene. That's quite interesting. You have the purity over or above 99.9%, and no need to cyclization and aromatization reactions. And only a few fractions need to do the hydrogenation. That is just to show the intensified reaction system, the relatively small equipment that allows you to have very high conversion, high selectivity, in the lower resistance time, as well as the intensification purification system. I mean, allow us to get over 99.9% of purity. So that's high purity, very low freezing points, below 47 degrees centigrade without any kind of additives. So some final comments. Jet fuel rods are available. So, and then you are able to obtain jet fuel and renewable hydrocarbons. Many roads for that. But economic feasibility is a barrier. That means that you have to do economic assessment and life cycle analysis to discriminate between possible roads and feedstock. Many roads are available, as I mentioned to you, and they have the impact of raw material costs that can be very significant together with the logistic, especially if you take account that the biomass is region dependent. For that, the process that's flexible is quite important. Just to finish, I would like to mention the economic viability is essential. Society in general is not prepared to pay more because it's renewable. And the criteria of circular economy have to be met in this process, and you have to be able to deliver to the society a biofuel, including jet fuel, that are competitive in terms of price and friendly with environment, so that it could be accepted for the society. I would like to thank you for all the students, researchers of LOFC and DPS, especially Dr. Battistela, Dr. Nívia Lima, Professora Maria Regina Maciel, that are co-authors of the Unicamp Biokerosene process patent. Thanks very much.
Hello, my name is Thiago Olitta Basso. I'm an assistant professor of the Department of Chemical Engineering at the University of São Paulo. Title of my presentation is Physiology and Lipidomics of Saccharomyces cerevisiae at extremely low oxygen availability in the absence of ergosterol and unsaturated fatty acids. We're interested to study extreme oxygen limitation because this is really important when we're looking to industrial scale bioprocesses. Due to the problems with mass transfer, it's common sense that such environments are virtually full anaerobic. As opposed to this condition, when we're studying at laboratory scale using bench scale bioreactors or chemostats that are considered a perfect mixture system, is not very always simple to devise strategies to minimize dissolved oxygen into the system. So it's not easy to study what are the consequences of anaerobicity in fermenting cells, in producing cells, microbial cells, or even animal cells. Therefore, for that purpose, we're interested to see what are the consequences for extreme oxygen limitations to microbial cells, in this case using yeast as a model organism. For that, we used this experimental setup, a bench scale set of bioreactors, running as a continuous cultivation, more precisely as a chemostat, and the system were equipped of tubings that are impermeable to oxygen. This is the scheme of how we study the transition from aerobic to anaerobic condition, because we want to test the normal saying that anaerobiosis can be easily achieved in a lab by using a hermetic bioreactor flushed with nitrogen gas. For that, we use the following strategy. We start cultivation using a defined media, injecting compressed air as the aeration system, and as soon as we achieve this steady state, we stop sparging compressed air and we turn to nitrogen gas, the industrial nitrogen gas that can contain up to 50 ppm of oxygen. And we were expecting a washout behavior of the cells, because when we do not add these anaerobic growth factors, what are the consequences? So if we look to the sterol biosynthesis in yeast cells, it started with isqualene and goes up to ergosterol, which is a very important esterol, such our cholesterol in our membrane, so in the yeast cell membranes. And for that, 12 moles of oxygens are required for this synthesis. The same holds for unsaturated fatty acids. The biosynthesis of unsaturated fatty acids, such as oleic acid, requires molecular oxygen. So when we don't provide this, the only way that cells can synthesize both ergosterol and unsaturated fatty acids is via scavenging this oxygen if this is present in the system. So we look to the kinetics and to the behavior of the cells during these transitions, and I want to point you to this. So if you follow the biomass, we had a steady state and then switched the gas to 50 to nitrogen. We could see a washout behavior, but to our surprise, what we found is that Saccharomyces cerevisiae did not wash out, and a new steady state was achieved at a lower but a steady state with 0.42 grams per liter when we use industrial grade nitrogen gas without providing this anaerobic growth factors, twin, which is the source of oleic acid, and ergosterol. So we said, okay, let's try a more extreme condition. So let's make it more anaerobic. So let's try to make it more difficult to cells grow. So we use the same setup. We started with compressed air, and then this time we shifted to ultra pure nitrogen, which is 10 times more expensive than a normal gas because of the purity, and we also add an oxygen trap. With this, the concentration should be in the range of 0.05 ppm of oxygen. So we're trying to suffocate these. And again, this is just the detail of the trap. So it was positioned before the entrance in the bioreactor. It's a commercial oxygen trap. And to our surprise, Saccharomyces cerevisiae SNPK, which is a lab strain, also did not wash out during this condition, and nil steady state was achieved even using ultra-pure nitrogen. We looked to an industrial strain known as PE2, but bioethanol strain, and it also did not wash out, achieving a biomass yield. We can see the major physiological parameters here that the industrial strain achieved slightly higher biomass during steady state. You can see here also the biomass yield, producing less glycerol when we look at the specific rate, and with a lower residual glucose concentration. And we said, okay, let's look to the profile of the lipidomics. So let's start with the sterol. So if Saccharomyces is scavenging O2 to grow to some extent, how is this affecting lipid biosynthesis? So here I show you during the aerobic steady state, we see an accumulation of ergosterol that requires all that amount of oxygen for both strains. That's normal situation. But when we deprive cells with oxygen and we don't add this ergosterol and oleic acid, the anaerobic growth factors, there is a completely shift to accumulation of iscoline and lanosterol, which is the first intermediate that does not require oxygen, and the second intermediate that requires only 1 mole. So there is a completely shift of the lipids profile in the terms of the esterols. And we also run an aerobic condition where we added these anaerobic growth factors, and we can see the similar behavior, so accumulation of ergosterol. We look at further to fatty acids, and very interestingly, normally cells accumulate unsaturated fatty acids. You can see here C16 and C18, both in aerobic and anaerobic conditions. You can see here. But when we deprive them of the anaerobic growth factors and we use that extreme oxygen, there is an accumulation of saturated C16, saturated C18, and shorter chain saturated as well, C12 and C14. We also looked to the important phospholipids and others such as ceramides and cardiolipin. So when we look to cardiolipin, which is an essential component of the inner mitochondrial membrane, we can see that cardiolipin is present both in aerobic as well as anaerobic conditions, but is completely absent when we deprive them of oxygen and these growth factors. And this is very complicated, problematic, because mitochondria is very important even in fermenting cells. I also would like to show call your attention to the ceramides. So ceramides regulate cell proliferation, and it's involved in apoptosis or programmed cell death. And you can see here there is a huge accumulation of ceramides in these conditions, higher than the anaerobic and much higher than the aerobic, and this is can be very lethal to cells. It's a deregulation. This compound is linked to various types of degenerative diseases such as cancer, diabetes, and very others. So it's a mess to cells. To check this, we expose these cells to 2 stresses, ethanolic stress and pH stress, and we could see that these cells were much more sensitive to such stresses, because we can see here a sharp of the viability as soon as they are exposed to such stresses, both the lab and the industrial strain. I would like to use this opportunity to present another work of the group, which is regarded to the use of CO2 to produce fuels and chemicals. For example, if you have a source of natural gas that can contain up to 50% CO2, we were interested in isolating photoautotrophic microorganisms that could convert CO2 to fuels and chemicals. And we were interested in the lipid profile of such organisms. For that, the group sampled a estuarine region near the coast of Santos, state of São Paulo, more precisely in a mangrove area, and samples were enriched or selected under a high CO2 atmosphere concentration using a mass flow controller. And these environmental samples from them, we were able to isolate 3 microalgal strains that are 2 of them, chlorella and cinedesmus, are known to be tolerant quite towards CO2 concentration. And another strain was Parachlorella kessleri. So we evaluate these strains under shake flasks with different CO2 concentrations in the headspace, 0.04, which is a normal atmospheric levels, 5, 15, and 30. And we can see this similar profile and the maximum growth rate. All strains were able to keep a high growth rate, even up to 30% CO2. But when we look to biomass concentration, the strain Parachlorella kessleri was able to accumulate more biomass than the 2 other strains under such conditions, and we decided to further evaluate this strain in a more controlled conditions. So for that, we use a set of 2 bench scale bioreactors, the same equipment used in the previous work with yeast cells. But this time we adapted to photoautotrophic microorganisms, including a set of lighting equipment here. This is how the setup. So we control the inlet gas by providing a mixture of CO2 and air to provide 5, 15, and 30% CO2 in the headspace. And you can see here the details of the light system inside the bioreactor. This is the apparatus. So when we look to the physiology and the lipid profile, we notice that this strain is able to grow up to 30% CO2. And although growth is a little bit slower towards the end of the cultivation, a 14-day cultivation, the growth rate during the exponential phase were pretty similar. The strain can keep a growth rate of 0.75 up to 30% CO2. And it can also maintain interesting biomass yield in the range of 1 gram per liter dry weight up to 30%. And very interestingly, when we look to the lipid profile, the major fatty acids, these 5 classes here, we can see clearly that as we increase the amount of CO2, there is an accumulation of or an enrichment of saturated fatty acids or monounsaturated fatty acids as compared to polyunsaturated fatty acids. And this is very interesting for high CO2 concentrations because both saturated and monounsaturated fatty acids are very important as biodiesel feedstock. So with this, I would like to concluding about the yeast work that although anaerobiosis has a clear absolute theoretical meaning in practice, anaerobiosis turns out to be not a clear-cut condition. Baker's yeast adjusts its lipid composition in order to grow within the amount of oxygen available. And regarding microalgae, the fatty acid varied in response to CO2 levels. High concentrations favor the production of saturated and monounsaturated fatty acids, which are suitable as biodiesel feedstock. I like to finish this presentation. Thank you very much for your attention. Thanking the team involved in the yeast work and involved in the microalgal work and the funding angels. Thank you very much for your attention.
Olá a todos, eu quero inicialmente agradecer a oportunidade e o convite da plataforma Biofuturo para estar participando deste evento. E no dia de hoje nós vamos falar um pouco sobre os biocombustíveis, vamos passar pelo biodiesel, que é um patrimônio que o Brasil tem, e falar do que nós imaginamos para o futuro dos biocombustíveis e um foco muito importante da nossa fala sobre as matérias-primas, porque sem as matérias-primas fica praticamente impossível, né, nós fazermos o biodiesel. Eu estou como CEO da ECB Group, é uma holding que tem como foco investir na área de biocombustíveis e na área da energia renovável. Também com foco no investimento no agronegócio brasileiro, que é outro patrimônio que o Brasil possui. Aqui estão minhas credenciais, estou trabalhando com o segmento de combustível há mais de 20 anos. Entre algumas coisas que eu julgo importante que nós construímos nesses últimos 20 anos, foi a fundação da APROBio, Associação Brasileira de Produtores de Biocombustível, a qual estou como presidente do conselho. E outras tantas ações que fizemos durante esse período, todas elas ligadas ao mercado de energia renovável e biocombustível. Essa é a nossa estrutura do grupo, para quem ainda não conhece. A ECB é a nossa holding de investimento, e nós temos 3 áreas de investimento abaixo. Nós temos uma área que vai trabalhar com alimentos, que é a ECB Importação e Exportação. Nós temos a área de imóveis e temos a nossa maior companhia, que é a BBEST-Biofuture, a qual, além de ter operações no sul do Brasil, ser líder de mercado no Brasil na produção de biodiesel, também tem operações em São Paulo, Genebra, na Suíça, e um grande projeto chamado Omega Green no Paraguai, nossos vizinhos, que é um país que nos recebeu muito bem, e nós vamos falar mais sobre o projeto Omega Green durante a apresentação. Antes de nós falarmos dos biocombustíveis avançados ou os biocombustíveis da segunda onda, eu quero falar um pouco sobre o biodiesel. Na nossa avaliação, o biodiesel é o melhor programa de biocombustível que o Brasil já fez. É claro que nós nos beneficiamos com todo o aprendizado que o Brasil teve em função do programa do etanol na década de 70, então muitos desafios que o etanol teve, o biodiesel se beneficiou e com isso nós caminhamos mais rápido. O Brasil iniciou o programa, para quem não está lembrado, lá no ano de 2004, 2005, mas efetivamente no Brasil nós começamos a produzir e adicionar o biodiesel ao diesel em 2008, com uma mistura de 2%. Essa mistura cresceu rapidamente, chegando hoje a 13%, e com uma expectativa de nos próximos 2 anos nós chegarmos a 15% de mistura, por vários motivos, mas eu quero destacar aqui uns 3 ou 4 motivos. O primeiro ponto é porque o Brasil é um grande produtor de matéria-prima. Como disse no início dessa minha fala, a matéria-prima é fundamental para que a gente tenha o desenvolvimento da indústria, da produção e a utilização do biocombustível. Nós, também no Brasil, temos um mercado, que é um mercado importante, o Brasil é uma economia importante, e um mercado que ainda é importador de combustível fóssil. Então, quando o programa foi desenhado, um dos pilares foi e continua sendo a substituição do combustível fóssil importado por biocombustível, no caso aqui o biodiesel, produzido no Brasil. E 2 pontos super importantes são os pontos ambientais. O biodiesel, dependendo de qual matéria-prima nós utilizamos, ele pode chegar à redução de 90% da emissão de gás de efeito estufa. Então, realmente é uma energia limpa e renovável. E o programa também tem um aspecto social muito importante, porque ele, além de gerar emprego no interior do Brasil, distribuir renda, ele também reservou grande parte da compra da matéria-prima da agricultura familiar. E dessa forma, nós hoje somos o único programa de energia no mundo que incluiu as pequenas famílias produtoras de matéria-prima nesse novo mercado que se desenhava. Então, na nossa opinião, o biodiesel e o etanol, que são os biocombustíveis da primeira onda, são dois grandes patrimônios que o Brasil possui e tem ajudado muito a economia brasileira, agregando valor às matérias-primas oriundo do agronegócio e produzindo uma energia renovável, um biocombustível ou um combustível que polui menos. Quando nós olhamos para o futuro, haja visto que o Brasil já fez essa importante caminhada de desenvolver o mercado de biodiesel e de etanol, nós estamos vendo novos biocombustíveis sendo produzidos e utilizados no mundo. E aí eu destaco aqui o HVO, o SPK e seus coprodutos como a nafta e o GLP verde. O HVO, como nós chamamos aqui no Brasil, é o diesel verde. O SPK também é conhecido como SAF, é conhecido aqui no Brasil como Biokerosene. Esses dois biocombustíveis são avançados, eles possuem uma rota de produção diferente do que o biodiesel. O biodiesel nós produzimos através de uma reação química chamada transesterificação, e nesse caso do HVO e do SPK, nós utilizamos uma rota chamada hidrogenação. Ou seja, nós temos uma biorefinaria muito parecida com uma refinaria que processa petróleo. Nós injetamos o hidrogênio, temos as reações químicas, depois temos uma fase de separação e ao final nós temos 4 produtos que são o diesel verde, o biokerosene, os coprodutos, a nafta e o GLP. Qual, vamos dizer assim, um grande diferencial desses biocombustíveis? O primeiro grande diferencial é que são dropping, então nós podemos utilizar em qualquer percentual de mistura e também nós podemos utilizar ele em todas as tecnologias de motores existentes, ou seja, um caminhão de 1950 pode usar o HVO puro, um caminhão saindo da concessionária agora também pode utilizar o HVO puro. E no caso do biokerosene, é a única alternativa factível nesse momento para o setor aéreo colocar em prática o plano Corsia, que é o plano de redução das emissões a partir do ano de 2027. Atualmente, na sua grande maioria, as aeronaves já têm uma garantia que podem utilizar 50% desse combustível e empresas como a Boeing já anunciaram que a partir de 2030 suas aeronaves vão ter a capacidade de utilizar em 100% a biokerosene nos voos de longa duração e de curta duração. Nesse sentido, o nosso grupo tem trabalhado há mais de 3 anos para desenvolver o primeiro projeto de biocombustível avançado da América Latina, que é o projeto Omega Green, que eu apresento ele para vocês de forma resumida e muito rápida. O Omega Green é um grande projeto que o nosso time está trabalhando há mais de 3 anos. Esse é um grande investimento na ordem de $1 bilhão, que será efetuado no Paraguai. E nesse momento, uma empresa brasileira está investindo no Paraguai em função de que as condições para esse projeto lá são mais atrativas. E toda a produção que nós vamos ter no Paraguai já está comercializado com 2 super major oils, a BP e a Shell, isso é um dado público, e esse biocombustível produzido, que a nossa expectativa que seja a partir de segundo semestre de 2024, ele vai ser enviado ao hemisfério norte, determinado o local de consumo pelos nossos clientes. Lá nós vamos produzir no Omega Green o HVO, o SPK e os 2 coprodutos, a nafta e também o GLP. E nesse sentido, eu queria também comentar que essa indústria nova que está chegando, as biorefinarias, além de solucionar os problemas de transporte, produzindo o diesel verde, produzindo o biokerosene para o transporte aéreo, seja de carga ou de passageiros, ele também vai dar a oportunidade para que o setor petroquímico tenha matéria-prima verde para aumentar a produção dos polímeros verdes. Então, essa biorefinaria, ela atende vários mercados da química fina e por isso estamos muito animados. O nosso projeto vai produzir 20 mil baris por dia de biocombustível. Estamos localizados numa zona franca e, como já falei, estamos trabalhando para que em 2024, no segundo semestre, a gente inicie a produção. Falando sobre matérias-primas, da qual eu acredito que é a grande oportunidade que nós temos aqui na América do Sul. O Brasil e os países vizinhos têm a grande oportunidade de sermos, no futuro, o novo Oriente Médio, porém, de biocombustíveis. E por que nós temos essa oportunidade? Porque nós temos todas as condições de produzirmos muitas matérias-primas. Hoje, nós já somos líder na produção de soja no Brasil e se pegarmos a América do Sul, somos líder muito à frente de outros continentes e nós temos outras matérias-primas que podemos desenvolver, como carinata, canola, girassol, óleo de algodão, destaco aqui a pongâmia, a macaúba, que são espécies, na sua grande maioria, super adaptadas ou espécies que já estão num grau de pesquisa bem avançado e que nós podemos produzir aqui. Eu trago duas oleaginosas para nós conversarmos um pouco. A carinata é uma cultura anual, todo ano a gente produz ela no inverno, porém ela não compete com o alimento e ela pode utilizar áreas ociosas. Hoje a América do Sul possui muitas áreas ociosas no inverno, então a carinata é uma excelente alternativa para completar a renda dos agricultores e também, no que diz respeito ao uso da terra, ela se beneficia porque nós estamos produzindo mais na mesma terra. A pongâmia é uma aposta muito grande que nós estamos fazendo. Nós já assinamos com a Investância um contrato para a aquisição de toda a produção de óleo de pongâmia para os próximos 30 anos. A pongâmia é uma árvore originária da Índia que tem uma capacidade de chegar a produzir 4 mil quilos de óleo por hectare. Isso é uma produção importante. E o melhor da pongâmia é que ela vai ajudar a reflorestar muitas áreas degradadas e é possível manter a produção atual e introduzir a pongâmia. Por exemplo, em áreas degradadas que hoje nós temos a produção de gado para corte, para carne, nós podemos entrar com a pongâmia e aí nós vamos ter áreas que vão ter duas aptidões. Uma, a produção de alimentos, que é a carne, e outra, a produção de pongâmia, que vai produzir óleo vegetal para o biocombustível e também vai poder produzir o seu farelo, que pode ser utilizado na engorda de gado. Então, eu tenho defendido muito que a América do Sul tem oportunidades ímpar e muitas oportunidades para desenvolver a produção de matérias-primas. No que diz respeito ao projeto Omega Green, nós vamos utilizar lá 60% de óleo vegetal oriundo da soja, 35% de gorduras animais, oriunda de suínos, frango e bovinos, e 5% de óleo residual, o Luuk, ou óleo de outras culturas, como é o caso da pongâmia. Vamos ter todas as certificações necessárias para atender todos os países do hemisfério norte e, com isso, nós vamos ter uma produção da qual ela é certificada, da qual ela é auditada e respeitando todos os bons princípios de produção, respeitando o meio ambiente, tendo programas sociais e levando desenvolvimento para a região do Paraguai e para a América do Sul. Nós acreditamos que juntos, com os biocombustíveis, nós podemos construir um mundo melhor e mais verde e mais sustentável. Muito obrigado.